Hello viewers, this is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So today we are going to discuss about the salvation of the previous PhD entrance examination where the Hyderabad Central University conducted in 2020-21. So this is the paper contain 70 questions. So among these 70 questions, 35 questions related to Part A and 35 questions related to Part B. So this is about 2020-21 question paper. So 2020-21 conducted 2020 cycle. So Part A, the first question is, see here my dear friends. So the first question is, one second. Okay. Right. So the main bonds that occur in the secondary structure of proteins are, so that the bonds are hydrogen bonds is the right answer. The second question, cellulose is composed of D-glucose units which condense through beta-1,4 glycosidic bonds. Okay. So cellulose is composed of D-glucose units which condense through beta-1,4 glycosidic bonds. The third question is, which one of the following amino acids are involved in phosphorylation by post-translation modification? That is serine. Okay, serine amino acid are involved in the phosphorylation by post-translation modification. And the CN distance in a peptide bond, carbon-nitrogen, CN means carbon-nitrogen distance in peptide bond is typically 1.32 angstrom units. Okay, 1.32 angstrom units. The photosynthesis process is a, remember my dear friends, photosynthesis is reductive, endogenic and anabolic process. Okay, reductive, endogenic and anabolic process. Then next question, hypochromatic, hypsochromic shift causes the change in spectral position to shorter wavelength. That is shorter wavelength. And the seventh one is which of the following is not, remember here, not correct about plant viruses. So replicate by binary fission. So replicate by binary fission is not correct answer. So this is the right answer for this question. But actually the plant viruses are used precise gene editing and used in virus mediated overexpression and used in virus-induced gene silence. Okay, so these are all correct, but replicate by binary fission is not related to or not correct about the plant viruses. So this is the right answer of this question. Then the eighth question, in the process of protein transport to the chloroplast, which of the following class of proteins is more likely to lack a transit peptide? That is outer envelope specific proteins. The correct answer is outer envelope specific proteins. Okay. Then, right. The next question. Consider the following statements and choose the correct answer. See here, this is the question, assertion and region. So assertion, in plants chloroplast, MATK is considered a better DNA barcode than mitochondrial CO1. The reason, MATK is one of the most rapidly evolving coding sections of the plastid genome, which gives it good discriminatory power, whereas the sequence of the plant CO1 gene is highly conserved, thus limiting its discriminatory ability. So in this question, both assertion and reason are true. And the reason is, this reason is the correct explanation about the assertion or the above question. Then the next question, which of the following enzyme is used to remove 3' overhangs or fill in of 5' overhangs to form blunt ends? That is T4 DNA polymerase. Okay, T4 DNA polymerase enzymes is used to remove 3' overhangs or fill in of 5 overhangs of the blunt ends. Okay. Then the next question, which of the following enzymes is involved in the biogenesis of small interfering RNA in plants. That is RNA polymerase 4. RNA polymerase 4 enzymes is involved in the biogenesis of small interfering RNA in plants. Then the 12th question, Pell's blue staining is test used to demonstrate. So Pell's blue staining is used for the test to demonstrate of the ferrous or iron. Then consider the following statements and choose the correct answer. Statement 1, sucrose is the major transport sugar in the higher plants. Yes, that is correct. And statement 2, sucrose is a non-reducing sugar. So both they are correct answer. So both statements are true. Then in an ideal condition, when single-stranded DNA, ssDNA, is allowed to renal, how many classes of fragments can usually be distinguished by their frequency of repetition with a plant genome? That is 3. Okay, now? 3. 3 classes of fragments can usually distinguish. How many classes? 3 classes. 3 classes of the fragments can usually distinguish. Okay. The next question. If 20% of the nucleotides from DNA molecule contain the base T, what percent will be the base G? So, for example, if we know about the 100% of the purines and pyrimidines, here the T, so according to A always pair with T, Chargaff law, and G always pair with C. Here, my dear friends, so always if 20% of the nucleotide, that is a T contain 20%. See here. So T 20%. So then automatically, see here, my dear friends, T, that is 20%. Okay, T 20%. At the same time, A is also always definitely 20%. So 20 plus 20, 40% is over. Then remaining 100, 40% is over, remaining 100. So G 30% and C also 30%. So the containing, what percent will be the contain G? That is 30%. What contains, if the question is what percent will contain the base A? That is 20%. Okay. Base A contain 20%. And base C also 30%. Base C also 30%. Then the next question. Consider the following statements and choose the right answer. Statement 1. So statement 1, the enzymes responsible for the transcription in prokaryotic and eukaryotic cells are called DNA-dependent RNA polymerases. Then the statement 2. So RNA polymerase moves along the template DNA strands towards its 3' end during transcription. Here, only statement 1 is true. Only statement 1 is true. That is, the enzymes responsible for the transcription in prokaryotic and eukaryotic cells are called DNA-dependent RNA polymerases. So this is true, and whereas the statement 2 is wrong answer. That is false. And 17th question. The Mendel's dwarf pea plant was defective in the synthesis of plant hormone, that is gibberellic acid. Okay, gibberellic acid. And which one of the following molecules is the common precursor for the biosynthesis of carotenoids, gibberellic acids, and abscisic acid? That is geranyl polyphosphates or pyrophosphate. Geranyl 1 or geranyl pyrophosphate is the right answer. Okay. This is the precursor of the biosynthesis of the carotenoids, gibberellic acids, and abscisic acids. So these, among these carotenoids and gibberellic acid, and even though abscisic acids, precursor is geranyl phosphates. Okay, very, very important. Geranyl phosphates. And in the ABC model of the flower developments, in the ABC model of the flower development, we are observing the Arabidopsis thaliana plants. We are observed that is a combination of the specific petals. So petals should be AB. Okay, now, AB. Then which one of the following plants has both strategy 1 and strategy 2 for iron uptake in roots? That is in rice plants. Okay, rice plants shows about the both strategy 1 and strategy 2 for iron uptake in roots. Which one of the following hormones has its receptor localized on the endoplasmic reticulum? That is ethylene. Okay, ethylene hormone has its receptor localized on the endoplasmic reticulum, ER. This is one of the cell organelles of the all living organism. And transposable elements, we are also called the transposable elements, also we can call as jumping genes, like bobo mediated intrachromosomal recombination in Drosophila, such that two bobo elements in the same chromosome pair and recombine with each other. What would be the result of such recombination if the bobo elements were oriented in the same direction on the chromosome? That we can also called as that is deletion. Okay, now that is deletion. Next question. Mutation in a single nucleotide resulting in a codon that codes for a different amino acid is called, that is missense mutations. That is missense mutation. Okay, now single nucleotide mutation in occurred, single nucleotide resulting codon that is codes the different amino acid, we can call as missense mutation. Then 24th question. Suppose the average molecular mass of an amino acids is assumed to be 100 Dalton, Daltons. How many nucleotides will be present in an mRNA coding sequence specifying a single polypeptide with a molecular mass of 27,000 Daltons? That is, the right answer is 810. Okay, now right answer is 810. Next, if Darwin's syndrome occurs, syndrome occurs about in 1 to 700 in Turner syndrome, occurs in about 1 in 5,000 in the general population, and each is separated and randomly distributed in the general population, what is the chance that a baby will be born with both these abnormalities? What is the abnormalities? Down syndromes and Turner syndrome. This is Down syndrome and this is the Turner syndrome. So both the these abnormalities, how many gets chance? That is 1 by 35 lakhs. 1 by 35 lakhs of the population. Only one by 35 lakhs of the population get chance of the both the these abnormalities, Down syndrome and Turner syndrome. And which of the following is not an essential mineral nutrition in plants? Silicon. So silicon is not essential nutrients in the plants, especially. So 9 elements are the macronutrients and 7 elements that is micronutrients. Along with 7, the another one is eighth one is micronutrients, that is nickel. So nickel is one of the best micronutrients. If the deficiency of the nickel, we are observed mouse ear in pecan. In pecan plants, mouse ear disease, we are observed the deficiency of the nickel. Okay. Then, if an organism produces 16 types of the gametes of the organism, heterozygous is heterozygous for 4 genes. Okay, 4 genes. And if the only 4 types of the gametes, the organism is heterozygous for 2 genes only. Okay, if 4 genes, 16 types of the gametes we are observed. If 2 genes, only 4 types of the gametes we are observed in the heterozygous condition. And a set of virulence genes, that is we can also call as Vir genes, is located in Agrobacterium TI plasmid, tumor inducing plasmid. TI plasmid is activated by syringon, hetero or aceto syringon. Then, which mineral element is required for the synthesis of cytochrome C? So, cytochrome C, we are observed the element required for the synthesis of the cytochrome C, that is iron. Okay, ferrous. And in conventional approach of determining the genetic basis responsible for a known phenotype is called as forward genetics. Okay, forward genetics. The conventional approach of determining the genetic basis responsible for a known phenotype is called forward genetics. Okay, then the mutant phenotype can be rescued to wild type by genetic manipulation. This method is known as gene complementation. Gene complementation. The mutant phenotype can be rescued to wild type by genetic manipulation. This method is known as gene complementation. Then the 32, the set of all the genes present in a given specific species is called pangenome. Pangenome. Then match the following inhibitors. Okay, the correct pairing we are observed here, that is a transport chain complex. We are observed in the electron transportation chain, 5 complexes we are observed. So that is complex 1. So complex 1 represent rotinone. Complex 1 represent rotinone. And complex 2, that is inhibitor. That is complex 2, malonate inhibitor. And the third one is that is complex 3. Complex 3, antimycin A is acted as a inhibitor, competitive inhibitor. And complex 4, that is azide. Azide is the competitive inhibitor in a transportation chain, complex 4. And the complex 5 transmission inhibitor is oligomycin. Oligomycin is the inhibitor of the transportation chain in electron transportation. Okay, so this is the correct pairing. And the next one is, which one of the following is not related to enzyme kinetics? That is the Gatterman-Koch reaction is not related to enzyme kinetics. So remaining all related to enzyme kinetics. Michaelis-Menten equation, Hans-Wolff plot, and ED-Hofstee diagram. So these 3 are related to enzyme kinetics, whereas the Gatterman-Koch reaction is not related to enzyme kinetics. And the 35th question, identify the correct order of the basis molecules or modules, basis modules in a mass spectrometer. So that is ion source and focusing lens and analyze detector. This is the correct sequence. And the part B we will going to discuss about in this PhD entrance examination conducted in 2021 Hyderabad Central University, HCU. So that is part B, 36th question, red flower plants, that is the Mirabilis jalapa. So Mirabilis jalapa is also we can call as 4 o'clock plants. Okay, evening primrose also we can call as evening primrose or 4 o'clock plants, Mirabilis jalapa. So when crossed with the white flower, the flower produced by F1 plants where pink color we are observed. So when F1 individuals are self-crossed, the phenotypic ratio F2 plants will be, F plant will be 1:2:1. Okay, 1:2:1. That is 1 red, 2 pink, and 1 is white flower. Okay, 1:2:1. So this is the best example for the what we can say that incomplete dominance of the Mendelian principle. Our Mendelian principle not able to explain about the incomplete dominance. So that is 1:2:1, 1 pink, 1 red, 2 pink, and another one is white flowers. And then consider the following reactions that occur during glycolysis. Okay, glycolysis we are observed in the cytoplasm of the all living organism, whether the prokaryotic or eukaryotic organism. Emden Mera Paranas, EMP. So this glycolysis also we can call as EMP pathway. We know that. So in this, the following reactions occur in the glycolysis. That is So conversion of fructose 6-phosphate to fructose 1,6-phosphate. Fructose 1,6-phosphate. So here, this is the reactions are not reversible. In these reactions, which one is not reversible? So remaining are all reversible. Only 4 option is the not reversible reaction. Remaining are reversible reactions. That is conversion of glucose 6-phosphate to fructose 6-phosphate. This is isomerization reaction. So that's why it is reversible. And conversion of the glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So this is also reversible reaction. And conversion of 2-phosphoglycerate to 2-phospho-phenol pyruvate. So this is also a reversible reaction in the glycolysis. And whereas, so the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is not a what we can say reversible reactions. And consider the following statements, that is H plus ATP. ATPH is in higher plants. That is the true statements are 1, 3, 4 only. So 2 option is not true statements about the regarding of this question, and remaining 1, 3, 4 are the true statements. That is H plus ATP is used for energy of hydrolysis of ATP, and H plus ATP is maintenance the cytosol pH in the range of 7.3 to 7.5, and this ATP generates proton motive force. Okay, so the next question, this figure shows about this is A figure shows about the process underlying the biological transformation of nitrogen compounds. Match the correct with B. See here. So A is 4 option. So here, 4th one, nitrogen fixation. See here, this is the nitrogen fixation, and B related to that is denitrification, and this is E and F. E and F, that is ammonia assimilation and ammonification. Ammonia assimilation and also ammonification. So E is ammonia assimilation. F is here. F is ammonification. E means ammonia assimilation. Then C is nitrification. C is nitrification. And D is the remaining, that is nitrogen assimilatory nitrate reductase. Assimilatory nitrate reductase. So this is about the complete, what we can say that D is, my dear friends, D is assimilatory nitrate reduction. So among these questions, we are observed this is about the biological nitrogen fixation. So biological transformation or nitrogen fixation cycle. So this is the entire we can call as nitrogen cycle. Then next one, 40, transport of xenobiotics into plant cell vacuoles for their detoxification is facilitated by ATP binding cassette transporters. Okay, ATP binding cassette transporters. And the 41 question, ammonium persulfate used to polymerization of acrylamide and bisacrylamide. Acrylamide and bisacrylamide to prepare polyacrylamide gels for electrophoresis is, that is an oxidizing agents. So this is all oxidizing agents. Okay, now ammonium persulfate used for the polymerization of the acrylamide, bisacrylamide and polyacrylamide. Acrylamide, bisacrylamide and polyacrylamide gels for electrophoresis is an oxidizing agent. Okay, now that is ammonium persulfate used for oxidizing agent. Then the 42, for preparing 1 liter of 500 millimole disodium ethyl anidyne ammonia citrate estate, that is 2H2O EDTA. So that is disodium salt dehydrate molecular weight 372.24 gram per mole solution in water, and make up the volume to 1 liter. That is the first option is right answer. Then phase T7 promoter, bacteriophage T7 promoter containing plasmids are used for overexpression in cloned genes because of the execute specificity of T7 RNA polymerase to phase promoter. That is C option. Then 44. Macromolecule chitin is, that is nitrogen containing polysaccharide. Then the first step to two-dimensional gel electrophoresis generates a series of protein bands by isoelectric focusing. In the second step, a strip of this gel is turned 90 degrees, placed on another gel containing SGS, and an electric current is again applied in the second step. That is, the proteins with similar isoelectric points become further separated according to their molecular weight. Then 46, location of protein cleavage by trypsin, that is lysine and arginine, okay, amino acids. Then if 10 gram of sodium chloride, NaCl, FW, that is a 58.44 are dissolved into 100 ml of water, what is the molar concentration of the sodium chloride in the solution? That is 0.71. In competitive inhibition, Vmax unchanged and Km is increased. Very, very important, my dear friends. In competitive inhibition, Vmax is unchanged and Km only increases. In glycoproteins, the carbohydrate moiety is always attached through the amino acid residues, that is asparagine, serine, and R-threonine. The relation of D-glucose with D-galactose and D-mannose respectively is C4 epimer and C2 epimer. And which of the following statements about histone is incorrect? Means not correct statement. That is, they are negatively charged. This is the right answer of this question, but it is not related to histone. But histones are, they are bound tightly to the DNA. Histones are highly alkynyl proteins, that is basal proteins, and they can be modified to regulate expression from DNA. Okay. Then 52 question. What is the correct answer of this function of the nonsense codon? So that is, it releases the synthesized polypeptide chain from ribosomes. Okay. That is the correct statement about the nonsense codons. Then 53. In summer squash, white color fruit, that is W, and dominant over yellow color, that is small w, and disc-shaped fruit, that is capital D, and dominant over spear-shaped fruit and small d. Determine the genotype of the parents if the white color disc-shaped fruit crossed with white color spear-shaped fruits gives 3/4 white color, and disc-shaped fruits and 1/4 yellow color disc-shaped fruits. That is W, capital W, small w, capital D, crossed with capital W, small d, small d. And 54th question, recessive genes small a, b, c, d, e, and f are closely linked in a chromosome, but their order is unknown. 3 deletions in the region are examined. 1 deletion uncovers A, D and DE, another uncovers C, D and F, and the third uncovers B and C. What is the possible order of the genes? That is A, E, D, F, C, B. Then 55th question, the vector used in recombinant DNA technology or DNA technology should have many desirable properties. So that is rDNA, recombinant DNA technology. The vector is, that is the correct statements in this 1, 2, 3, 4. So 5th question is not related to above question. Only 4 options is correct statements. That is, it should be autonomously replicable or replicating. It should have a unique restriction enzyme site for various restriction enzymes to insert a foreign GNF. And it should be easily isolated and purified. And it should contain at least one selectable marker gene. So these four options are correct statements about this vector in RDNA technology. Okay, then 56th question. If two genes A and B are 12.0 map units apart, one expects to find, that is 12.0% of the recombinant gametes from a doubly heterozygous parent. And 57th question. Garden P is a diploid organism with 7 pairs of the chromosome, that is 2n is equal to 14. How many chromosomes and chromatids are present in metaphase 2 of meiosis? So metaphase 2 of the meiosis, we are observed 7 chromosomes and 14 chromatids, my dear friends. 7 chromosomes and 14 chromatids. And 58th question. A cross is made between two different strains in Neurospora crassa. It is a blue mold or green mold in Ascomycotina fungi, differing with respect to a specific gene for mapping its position to the centromere. The tetrads used from cross, capital A and small a, were isolated and classified at first division and second division segregation patterns. Which of the following tetrads represents the first division segregation pattern? That is capital A, capital A, capital A, capital A, and small a, small a, small a, small a. So this is the first division segregation pattern. And Skoog and Miller, this is related to plant growth regulators. In 1957, classical experiments conducted with tobacco tissue cultures have put forth concept hormonal control of organ formation. Which of the following growth regulators were used in these experiments? That is kinetin and IAA. KN means kinetin and IAA. Okay. Then the characteristic feature of the F1 plants population. Remember, all F1 population should be heterozygous condition or heterozygotes. And the percentage increases homozygosity achieved for each gene generation of backcrossing, that is 50%. Okay, 50%. Then the minimum size of the progeny population resulting from the random union of all possible gametes from the parents with the genotype capital A, small a, capital B, small b, capital C, small c is 64. And the proportion of genetic variance to the total phenotypic variance is known as heritability. Okay, heritability. The gradual loss of the variability from cultivated plants and their related species is known as genetic erosion. Genetic erosion. And the 65th question. In one of the following DNA markers, the application of the both polymerase chain reaction, that is PCR, discovered by Carey Mullis, and a restriction digestion of the DNA is carried to generate polymorphism in plants, that is amplified fragments length polymorphism. Okay, the 66th question. One student was working in a single nucleotide polymorphism in a plant species. The student was performing single nucleotide polymorphism, SNP. Typing used a technique called ARMS test, that is ARMS test or ARMS test. What is the full form of the ARMS? ARMS means amplified refractory or refractory mutation system. ARMS means the abbreviation of ARMS, amplification refractory mutation system. So this, what is SNP? SNP means single nucleotide polymorphism. Then 67. What is the most important characteristics of the shuttle vector? The shuttle vectors that can replicate the cells for more than one organism. Okay, very, very important. More than one organism. They can replicate more than one organism. We can call it as shuttle vector. The type of vector we can call it as shuttle vectors. Generally, during the polymerization chain reaction, PCR reaction cycle, what is the top lipid temperature for the PCR machine? That is almost 10 degrees centigrade greater than denaturation temperature. Okay. Then which of the following is C3 to C4 intermediate plants? That is Panicum vulgare. Panicum plants, which is the intermediate plant between C3 plants and C4 plants. The last question, the ovule that are completely inverted, so that the micropyle is facing downward and situated near the base of the funiculus are called, that is anatropous ovule. Okay, anatropous ovule. Okay, so this is the last question of this PhD entrance examination. The right answer of this 17th question is anatropous ovule. Okay, so I will stop here, my dear friends. If you have any doubt, please ask me. I will clarify your doubts.